Assalamu alaikum. The topic today is identification of viruses in cell culture. So there are several techniques and different methods to identify the viruses in the cell culture, and these techniques are chosen depending upon the severity and the severity of the viruses, that which virus have to be going to under observation. So we have several types of techniques to detect the virus in culture media. We have number one, cytopathic effect. Second one is heme absorption. Third is immunofluorescence, enzyme link, immunosorbent assay, polymerase chain reaction, and electron microscopy. So in the first observation, cytopathic effect observation, we have observed that many viruses induces the characteristic changes in the infected cells. It means that the cell that is infected includes cell rounding. Sometimes it get detached, and sometimes the fusion of cells occurs. And this is also, and these all the characteristics features can be observed under microscope, which indicates that the person having these characteristic features having a viral infection. And the second one is heme absorption. In this, some viruses infect the red blood cells, RBCs, erythrocytes, and when the RBCs are adding to the culture, so heme absorption occurs by the viruses. That indicates that the virus is also included in the blood cells. Number third is immunofluorescence. This method uses antibodies labeled with fluorescent dyes that bind specifically to viral antigens. And when the infected cells are then identified under a fluorescence microscope by the presence of fluorescence signal. Then the number fourth is ELISA, that is abbreviation of enzyme linked immunosorbent assay. In this, the enzyme linked antibodies produce a cholinergic change which when bind to a target. It means that when the enzyme linked antibodies bind to a viral sites, viral antigens, so it can cause a color change that indicate the presence of virus in the particular sample. Then the polymerase chain reaction PCR and reverse transcription PCR, RT-PCR. So in COVID pandemic, you have heard many times the PCR test. So the PCR test is basically the test for the detection of the virus in a culture. So in this, the molecular techniques are highly sensitive. These are the highly sensitive and specific for detecting viral nucleic acids. It means that this test is only the specific for the detection of viral nucleic acids. There are two types of viral nucleic acids in DNA and RNA. So the PCR is used particularly for the DNA viruses, and reverse transcriptase PCR, RT-PCR, is basically used for the RNA viruses. And number six one is electron microscopy. Basically, the electron microscopy is cost effective and requirement are too high, so therefore it is not get into practice regularly. Electron microscopy can provide direct visual evidence of viral particles in cell culture. And all of these methods had its advantages and limitations, and often a combination of methods is used to confirm viruses identification in cell cultures. That all about the viral identification in the cell culture.